everyone. Thank you very much for joining us in the second talk from the Physio webinar. This is an event organized by graduate students from the Physiology graduate program of the Institute of Biosciences of the University of São Paulo. This is our team. We organized 4 very interesting talks that will happen during October and November with speakers from different countries talking about different themes of physiology and their intersections with other areas. Please follow us to get more information and so you do not miss any of the talks. Today, we are very pleased to welcome here Dr. Raymond B. Huey, who graduated in zoology at the University of California in Berkeley. And he also got his PhD in biology at Harvard. He was a professor at the University of Washington since 1977 and retired in 2014, but he continued his research. Dr. Huey's research focuses on evolutionary questions involving the physiology, behavior, and ecology of ectotherms. He is especially interested in exploring patterns in the evolution of physiological mechanisms over different time scales. He has published papers related to global warming and thermophysiology in several ectotherms, including vertebrates and invertebrates. His current research studies the vulnerability of ectotherms to climate warming and how these animals deal with climate change. His analysis suggests that tropical ectotherms are especially vulnerable to climate warming, even though the rates of tropical warming are slower than in northern temperate and Arctic areas. Dr. Huey, thank you very much for your presence here today. After your talk, we'll have 30 minutes to take questions from the audience. Everyone can ask questions through the YouTube chat. They can also be in Portuguese if you like, and the team will translate for me, and our team will select them and send to me. This event will also be recorded and will be available through YouTube after the transmission. With no more details, I give you the word now, Dr. Huey.
Can you see that?
Yeah, we can see it. It's perfect.
Perfect. Okay. Well, thank you, Stephanie, and thank you very much for the invitation to spend this part of this afternoon with you. I really wish I could be there with you in São Paulo, but unfortunately, it's the times. But I do want to thank the graduate students very much for this invitation. It is a special honor to be invited to give a talk by graduate students, so I appreciate that very much. And because the talk is an invitation from graduate students, I thought I should start with my own time as a graduate student and explain how I became interested in thermal biology, and then give several examples of recent work that I'm doing. Okay, if you go back more than half a century ago when I started graduate school in the late 1960s, and asked what were the really hot areas in terrestrial ecology at that time. And these were the areas that I found exciting and my friends and professors were studying. Physiological ecology was not really part of that. Certainly there was work being done, but it was not, at least in my world, at the forefront of contemporary ecology, certainly not the way it is now. So how did I become interested in this field as a graduate student? Well, there's several things, but the primary one was that in 1969 and 1970, I had the great good fortune of spending 10.5 months in the field in the Kalahari Desert in Southern Africa, working on a series of sites. And I camped out for that time So I experienced the full seasons. I experienced summer, I experienced winter. And the Land Rover that we had did not have a heater or an air conditioner. So we lived the seasons for almost a full year. It was a very exciting place to work. There were some spectacular lizards. Incidentally, this is Eric Pianca, who led the trip. This is me. Yes, I once had black hair. And Larry Coons. So it was a fantastic place for a young biologist to live in the desert for so long. And there were some other animals there that made walking around in the desert looking for lizards exciting. But over the course of months of being in the desert and experiencing the seasons, I learned very quickly that what determined where and when lizards were active was not so much competition and predation, but it was really the thermal environment. And here's one example for an agamid lizard. In the summer months, it's active very early in the morning and late in the afternoon. During the heat of the day, it's down a burrow. In the winter, it's active only during the middle part of the day. So where and when those lizards were active was easily predicted simply by thinking about the temperature at that time. When I left Africa and restarted graduate school again, I started working in the Caribbean, in particular on anolis lizards. And one of the projects involved looking at these lizards in an open habitat, and they live on these tree trunks and they can shuttle between sun and shade very easily. And just a few meters away is a deeply shaded forest. And I looked at the body temperatures of those lizards in the open and the forest, and they were very different. They were thermoregulating carefully in the open. They were thermoconformers in the forest. And one of the questions that came up when I was doing this work was, what does it mean for a lizard to be at a temperature of 32 versus 24 or 25? Did that have any effect on their ability to capture prey or escape predators or perhaps interact socially? And that's what I wanted to try to explore. And the idea that I came up with was to be inspired from field data, but to go into the laboratory and then take a trait and measure how the performance of that trait changed as a function of body temperature. And if I could do that, then if I knew that a lizard was active at this temperature, I could predict its performance. A very simple idea. The first question was, what performance measure should I use? I wanted a whole organism trait, something that was directly relevant to their ecology. And so I decided probably a good trait would be locomotion, speed and acceleration. That would be important for a lizard to capture prey or to escape predator, or in this case, to avoid being trampled by a bison. If you can't cross that field in 9 seconds, you shouldn't go to that field because the bull can do it in 10. Okay, so that was the basic idea. But this is the early 1970s. How does one measure acceleration and sprint speed in something like a lizard? Microcomputers did not exist. And I was in an ecology lab. We had no physiological equipment. So trying to measure locomotion was tough. But I had a pet frog. I had a meter stick. And so one day, in the basement of the biolabs at Harvard, I would take the frog, change its temperature, and see how far it could jump. Then I just fit a simple polynomial, and that enabled me to estimate the optimum temperature and whether it was a generalist or specialist, or I could predict, you tell me the temperature, I could predict how far that frog would jump. So that was my first thermal performance curve when I was a grad student. But I really wanted to measure locomotion in lizards, speed and stamina. And when I was a postdoc at Berkeley, I worked with a fellow Miller postdoc there who is an electronics genius, and we designed basically a clock apparatus where I could put a lizard in a track, chase it towards a simulated burrow at the end. And there was a series of photocells down the track. And when the lizard would break that beam, that would be recorded by the clock apparatus that we built, and we could measure speed. So we have the field data. Now I have how fast this lizard can run as a function of temperature. And this enables me to map performance at different times of day. And this is basically what I was able to show. So in this environment, which is in lowland Puerto Rico in summer, even at sunrise, the temperatures are pretty good, pretty warm. So the lizards are able to run at 90% of maximum speed at sunrise. Okay. Other times of year, this would not be the case. Okay, so this is the early 1970s, probably 1973 or 74 when I did the frog jumping. And I had this idea that you could measure a thermal performance curve and you could use that to explore what temperature meant in nature. You could ask a whole series of evolutionary questions about the evolution of performance curves. It was obvious that this was a useful idea. So I was feeling fairly proud of myself. That was my first sort of really good idea in biology, and I was pleased with it. Until I read a paper by J.R. Brett. It's published before I did the frog jumping. I didn't know about this. It was probably a beginning postdoc when I discovered this paper. My first response to it, once I got to page 2 or 3 into the paper and saw this graph, was major disappointment. I was really crushed. Let me blow up this plot and show you what it's like. Brett was studying growth rate of salmon as a function of both temperature and food. So this is a plot, it's growth versus temperature, positive growth, negative growth. If you give them all of the food they want, this is the curve you get. As you lower the food ration, the curve changes shape dramatically. Notice that the highest temperature at which growth is possible gets less and less as you lower the food ration. In other words, growth becomes more heat sensitive as you lower the food level. That's pretty wild. Moreover, the optimum temperature is shifting dramatically, such that the optimal temperature, if the fish has very low food, optimal temperature for growth is at a much lower temperature than if it has lots of food. This was pretty crushing because what I realized is that there was not a performance curve for growth. There was an infinite number of them. And that meant that my simple idea of mapping temperature to performance in nature was more complicated than I'd realized. And that's why I was so horrified to see this graph. To realize my idea was way too simple. Life is more complicated than that. So initially I was pretty unhappy when I read this paper. But I kept coming back and looking at this plot and saying, boy, that's really an interesting set of curves. It's not just that growth drops uniformly at all temperature, it's shifting, the curve is shifting. And so I started trying to think of what could be a physiological explanation for this and came up with a very simple energetic model where growth is the difference between how much energy an animal can take as a function of temperature, but then there are metabolic costs associated with digestion and just routine maintenance. So growth, the ability to grow, depends on the difference between these two curves. In other words, the net energy gain. And we can plot that now as net energy gain as a function of temperature, simply the distance between these two curves. And now if we lower the food ration uniformly at all temperatures, we basically recreate Brett's curves. Okay, so now I was happy again. And was published this in 1982. There was a similar version published a few years before that by Reed Hainsworth and Larry Wolff, but it's this very simple energetic analysis behind Brett's data. So that was decades ago, and it was basically an issue of academic interest, but then climate warming started to become an issue. And at one point I thought, I wonder if Brett's findings and climate warming together had some interesting implications, and decided to explore that. And so I invited Joel Kingsolver, who's a long-term friend and a great colleague, to collaborate with me on this, to develop a relook at Brett's patterns, but to do so in a variable environment so we can manipulate temperature and explore that. Okay, so we set up a simple growth equation which deals with how is net energy gain at each temperature affected by both the food in the environment and processing. You need to know the distribution of body temperatures, and then you can solve. I'm not going to go over the model in any detail. It was published in November of last year, but we explored several possibilities for thermal generalists, for thermal specialists that have a narrower curve. We had different Q10s, different sensitivities of metabolic rate, and a variety of different body temperature distributions. We also had various levels for food. We could uniform drops at all temperatures, or one case where we had food becoming unavailable mainly at high temperature, as if food foraging, say, were restricted at high temperature. We explored all of these different combinations in the paper, and I'll just show you one example where what we're doing here is shifting mean environmental temperature and looking at net energy gain, high food, intermediate food, low food. And this is the case where food is available primarily at high temperature, but drops off at low. So it's very similar to what Brett found and provides a simple model for explaining how the thermal sensitivity of energy gain should vary with temperature and food. Why is that relevant to climate warming? Okay, here's a simple graph where I have now just net energy gain as a function of temperature. So the optimum is here. And let's assume before climate warming that lizards are normally active at this temperature, range of temperatures. That's great. They're active at temperatures at which they grow quickly. Climate warming comes along, that's going to force them to a higher body temperature, which in this particular example will lead to reduced growth. But if food is also declining at the same time, the optimum temperature for growth is going in the opposite direction. So animals should select a lower body temperature if they're given less food, but the environment is getting hotter and hotter. And we call this metabolic meltdown. And I'll explain this a little more in just a minute. But it basically says that if climate warming occurs and if food resources are restricted at the same time, that the effects of climate warming are going to be much greater than if it was just the temperature change alone. First question, will there be reduced food? Well, there can be reduced food for a variety of reasons. Climate change, habitat, pollution, a series of things. One of my favorite examples, empirical examples, is something called the windshield effect, where people in Europe over years have driven their cars on a particular stretch of the highway and then counted the number of insects that hit the windshields. And they have this back for decades. And here's one recent example published last year. This is a statistical analysis, so these are residual numbers. But what you can see is that the number of insects hitting the windshields have been declining over time. There are fewer insects there now than there were in the past. Climate warming can have direct effects. Whoops, where's we? On food. High temperatures in some cases will be deleterious to insects. You can get plant dieback Barry Sinervo has been showing this as reducing shade that's available to animals and also the food for insects and lizards going extinct on areas through this mechanism. High temperatures can also affect foraging activities. This is something that Barry Sinervo and colleagues in a major paper published in 2010 in Science have emphasized. And I'm going to focus on just one of these, how climate warming affects foraging time. So we were asked to write a perspective for the paper that Barry et al. Wrote. And I'll just show you a cartoon that we developed for that perspective. So the game is to try to predict when lizards can be active in the environment. You need to know three things. What's the hottest temperature in the environment? So if the animal goes and sits in the full sun, what's its temperature will be? The lowest temperature, operative temperature in the environment, which would be in the deep shade, the coolest spot. So that's the range of potential temperatures that an animal can have on the surface. Then we have to have a behavioral index of the temperatures that animal will accept for activity. Knowing that, we can predict when and when they're going to be active, and that's here. And at this time of day, temperatures are within this range, and in late afternoon, there again, environment cools down enough for activity. So now let's see what happens with warming. The lowest temperature is hotter and the highest temperature both go up. And now the activity time is reduced. There's no longer activity in the afternoon because it's still too hot, and the morning activity period is reduced. This means that the lizards have less time to forage. If they have less time to forage, they're probably going to get less food. If they have less food, then by Brett's effect, they should select a lower body temperature than they are here. So let's say rather than going 38 to 33, they shift down 35 to 30. Well, if they do that, now the only time that they can achieve those temperatures reduced even more. So the behavioral shift makes things worse. And that's why we're calling this metabolic meltdown. That if you have climate warming, even without a change in food per se, but simply get a restriction in the time of activity, it will lead to a further restriction in activity time, which will reduce food, which will basically snowball to extinction. Okay. So this has been a fun project for me because when I first saw this figure many years ago, as I said, my first response was horror. I really didn't like it, because I saw that life was much more complicated than my simple models. But fortunately, I stuck with it and I kept looking at this figure, trying to figure out what the mechanistic basis was. And then decades later, working with Joel, finding that this effect would be relevant to climate warming. And we have an expression, turn the lemon into lemonade. And Carlos says we have one in Portuguese as well. But this is a suggestion that as you go through your careers, you will inevitably find things that are disappointing. An idea is shot down by a paper or someone. And this is always disappointing. But sometimes you can find a way to turn that around, and what appeared to be a lemon can be a lemonade, or even better, caipirinha. Okay. So that's warming conditions. Let's turn now to a second topic, which is winter. And I'm going to talk about depth, the depth into the soil that animals hibernate. If you asked me a few years ago what I thought about winter biology, as a field biologist, this would be my answer. It's boring. It's cold. Nothing is happening. Everything is underground. It's just a boring time of year. I will admit that it has some really interesting physiology in terms of freeze tolerance, freeze avoidance, and similar kinds of things for very high latitude, high altitude animals. But in most parts of the world, temperate zones, winter biology is boring. I held that view for my entire career until a few years ago when I read another old paper, this one written by Raymond Cowles, written in 1941. And I'll explain in a few minutes why, when I read this paper, I became very excited about winter biology. Okay. But I have to have a slight detour, tangent as it will. Around 2002, Donald Rumsfeld was the Secretary of Defense of the US government, very high position. He was asked a question about what do we know about some country or some political problem in the world. This was Rumsfeld's response. He said, there are some things we know. These are called known knowns. Like, I know how to speak and read English. I know it, and I know it. There are also things that he called known unknowns. Things we know we don't know. There is a language called Mandarin. I don't know how to read it. I know I don't know how to read it. That's a known unknown. But there are also things which he called unknown unknowns. These are things we not only don't know, we don't know that we don't know them. We're unaware of their existence. When this quote first came out on the news, most of us laughed at Rumsfeld and thought, oh, this is really silly. But I found myself intrigued by the concept of unknown unknowns and realized this. I think much of science is studying known unknowns. We know that there's a gap in the field, and we explore to try to answer questions that we know. But I think some of the most exciting parts of doing science is when you discover something that you didn't know was an issue before. It's an unknown unknown. And for me, this desert work was really an unknown unknown. And that's what I want to explore. Incidentally, there is a field of psychology and epistemology about this whole area. It's called the Johari window. So it is an important part of knowledge. Okay, so let's go back to Cowles. In 1938, Cowles spent about a month camping out in the deserts of Southern California in winter. And he started walking around the sand dunes, okay? The little hummock dunes. Here's where he was working. Here's another view. Wait a minute, what's that in the sand? The snake tracks. Snakes active in the middle of winter? This observation intrigued Cowles, and during his time in the desert, he started thinking about what life is like for animals that are hibernating. He knew that they were spending most of their time below the surface of the sand, and then sometimes would come out and be active. And he asked two basic questions. How deep do they have to go to keep from freezing? Because it gets very cold in this environment. And if they're very shallow, surface temperatures can be freezing, can go below freezing and potentially kill those animals. But he's also interested in how do they know it's a warm day on the surface? How do they know that it's safe to come out and that they'll be able to bask and warm up? When I reread this paper a few years ago and started seeing this, I said, wow, this is really interesting. 8 decades ago, this was a long time ago when Cowles wrote this paper, he was asking questions about trade-offs the time no one else would do it. I don't know of any example of trade-offs, and I'll explain those in just a minute. I realized that there are a couple of other questions we needed to ask that Cowles did not ask. One is, what depth minimizes cumulative metabolic expenses over winter? If a lizard goes down and spends 4 or 5 months underground, it's going to use a lot of energy. What depth in the sand minimizes that rate of expenditure so it doesn't starve during the winter and might have some surplus in the spring? And the other question, why even come out at all? Why come out on a warm day? Okay. So I read that paper. This was the view. Within a few minutes of rereading that paper, I decided winter is actually really exciting and started exploring these questions and invited some good friends and colleagues, Michael Carney, Ophir Levy and Liang Ma, to work on this. And the work that I'll be talking about is in press in Ecology Letters, so it should be out very soon. So if one were going to explore questions like this, the first thing I would do, first thing I tried to do, was to go to the literature to find out what's known about the depth where lizards go, what's known about why they come out, and so on. And the first thing I found is that there's essentially no information whatsoever on this. And Cowles was asking these questions 8 decades ago, and there are no data. So in the absence of empirical data, the obvious approach is to use simulations to try to get some approximate answers into what might be going on and explore that. So what we did was to find a number of sites in the United States where there are soil temperature records at a series of different depths every hour for multiple years. And I pruned the data set down to 90 different localities that had very complete data at all depths for these 2 years. Are these localities relevant to lizards? I think so. Here I've downloaded all of the known localities for lizards in the United States, shown in red. Black shows our sites. And the median distance from one of our soil temperature sites to the nearest collecting sites for lizard is 28 kilometers. So I think most of these sites are relevant. Okay, let's look at how temperature changes in the soil, going from 5 centimeters down to 50. The patterns are not surprising at all, very obvious. There's much greater fluctuations near the surface. And if you do density plots or ridge plots for these, you can see that at a depth of 5 centimeters, there's much more variation in temperature. It gets much hotter and gets much colder. Okay. So first question is, what depth does a lizard have to go to avoid risk of freezing? Okay. Cowles predicted shallow depths. So here are ridge plots now. So each row here represents a month from January. So winter, summer, winter. This is 5 centimeters, 20, and 50. And no surprise, temperatures are much more variable in the shallow and much more stable down below. In theory, it's very easy to predict how deep the lizards have to go to avoid freezing. Let's say that the temperature that would kill a lizard is freezing temperature, just below freezing temperature that lizard tissue freezes. You'd see that if they were at 5 centimeters, that in the month of January and December, they risk freezing. But if they're at 20 centimeters or 50, no problem. They can go the entire winter with no risk of freezing, this is what Cowles expected. We can do this geographically. Here I plotted for the month of January for a depth of 5 centimeters at each site, the proportion of all of the hours in that month that were frozen. If it's red, that means that it never froze during that month. If it's increasing blue, means a higher proportion of the hours were frozen. And it's pretty clear pattern that mainly in the southern United States, there's no risk of freezing, but you get to the mountainous areas and high latitudes, there's a much higher risk of freezing. We can do this with a small multiples plot for all of the different depths. And you can see that if they get down to around 50 centimeters, no problem freezing anywhere in the States, almost anywhere. Okay. So that's the approach, but immediately one runs into some problems. Here I've used freezing temperature as the lethal temperature. But is that really a lethal temperature in nature? We don't know. mean, here are the standard ways that people go about measuring cold tolerance in lizards. The most common is a critical thermal minimum, but they're also freezing, supercooling, and survival times. Let's look at how a CT-min critical thermal minimum study is done. You start with a lizard at a warm temperature, you cool its body temperature rapidly, and eventually it loses its righting response, it can't turn itself over. That's the CT-min. Another approach, much less common, is a lethal temperature. You take an animal, you drop it quickly to various low temperatures, leave it there, and see how long it takes before it dies. Okay. So there are lots of CT-min, there are a few of these and a few of these. But I'm not sure that any of them is relevant to predicting depth, surviving depth in the field. Why? If you look at the cooling rates that are used, and this is from a recent survey of 51 critical thermal minimum studies, the median cooling rate was 1 degree per minute. So it's a very fast drop in temperature. Let's see what happens to the drop in temperature in the autumn at various depths. And it's easiest to see here. It's dropping 10 degrees over several months. So it's a very slow rate of cooling. At its shallower depths, it's cooling, but it's also having major fluctuations in temperature. It's not obvious to me that a measurement that's taken with a cooling rate of 1 degree per minute is at all relevant to predicting how cold is too cold for these animals. So the basic problem is this. We can easily measure soil temperatures and know what temperatures are available, but we really don't know what temperature to use as the lethal temperature. Here I've shown supercooling, freezing, and critical thermal minimum. This is an average for lizards. You get very different answers depending on which one you use. And my suspicion is none of the above is really relevant because given a very slow rate of cooling in the autumn, there's plenty of time for acclimation, but there's also plenty of time for cumulative damage. Maybe long-term exposure to a moderate temperature is damaging to lizards. So we have no idea really what is the what the lethal temperature is, or even how to measure it. It's very, very hard to predict. And in the paper, we talk a lot about various ways to try to approach that. Another question is, what depth has the lowest cumulative metabolic rate? This would be important for an animal surviving, being able to survive through the winter and have some energy left when it emerges in the spring. Here again are soil temperature plots from 5 centimeters down to 50. We can take those, we know what the temperatures are, we can see how temperature mix, these are reversed, sorry about that. Metabolic rate is a function of temperature, and then predict over the winter which depth minimizes metabolic rate. And if you do that, the depth that has for the highest percentage of sites that show the lowest cumulative metabolic rates are at 5 centimeters. These are only the sites where it didn't freeze. For other sites, they'd have to go lower, but it's still shallow is better in terms of minimizing metabolic rates. So there's an interesting trade-off. Being shallow runs the risk of freezing, but if you don't freeze, you have the lowest metabolic rates over winter. What about activity? Here I've plotted soil temperature on 2 different days at 5 centimeters, 10, and 20. And this is time of day, this is night, day, night. You can see what happens. Soil temperatures are cooling, sun sets early in the morning, and you get a rise in temperature, starts faster and earlier at 5 centimeters. There's a delay of an hour or two at 10 centimeters and a delay even further at 20 centimeters. So which depth maximizes the opportunity to be active? Problem for a lizard, if it's underground, what's the cue that it gets that it's a warm day on the surface? In winter, not every day is warm and sunny. Most of them, in fact, are not warm and sunny. So it would not be safe to come out. How do they know? Cowles was not very explicit, but basically he was suggesting that lizards had sort of a threshold temperature. And if the soil temperature and hence the lizards crossed that threshold, then they would come out and check out to see if it was a sunny day. So let's look at the thresholds at these time of occurrence for these depths. 5 centimeters, it comes here, a little bit delayed at 10. Here at 20 centimeters, it gets the signal, but it's nighttime. So it's no longer warm on the surface. Very clearly, the best place to be is shallow because you get the signal early in the day and you could come out, bask, and take advantage of that warm day on the surface. So that was basically Cowles' idea. Really interesting. Okay, so we can now formalize this using niche map R, a biophysical model developed by Warren Porter and Michael Carney. And we can predict whether or not lizards would be active, could become active depending upon the depth that they chose. So we need a threshold here. We're setting it 20 degrees. If the soil temperature crossed 20 and it's crossing at a certain fast rate, the lizards could come out to the surface and check things out. Here are the plots. This shows body temperature, predicted body temperature at 2.5 centimeters all the way down to 100. And it's obvious that if a lizard is at a depth of shallow depth of 2.5 centimeters, that it gets the signal many times and can become active in winter. If it's down 10 or 15 centimeters, that threshold is never met and it stays underground. Okay, so this is really interesting and it's in accord with what Cowles was predicting, but we're making a big question here. What cue do lizards use to detect that it's a warm day on the surface? Are they using a threshold of 20 degrees? And that would be shown here, blowing up part of that plot. Here, the soil temperatures are not reaching 20, but on this day, it did. They become active and come out to the surface. Do lizards do that? Nobody has a clue. No one has ever asked this question. What cue do lizards use to emerge in the middle of winter? We have a little bit of data from other seasons, but basically this is an unknown topic. And so I think it's a fun example in that it shows that when you're using a computer simulation, you have to specify certain parameters, certain rules to make those predictions. We just guessed and arbitrarily used 20 degrees. It might be 25 degrees. There might be a completely different set of cues. We don't know. But what this does is open the opportunity for studies where someone could go in and see what lizards are actually doing. What kinds of body temperatures bring them to the surface? You could use neurophysiological studies to do that as well. The final issue is, why are lizards and snakes coming out in the middle of winter? It seems risky. They're going to be cold when they first come out. There are birds and mammals that will be there that could prey on them. And it's not clear why they're ever coming out. Cowles curiously didn't address this, but I think it's an interesting set of questions. One is they might just be coming out to feed, to replenish energy stores. We have data from the Kalahari that suggests that this is the case for Kalahari lizards that are active in winter. But we know in some other populations that's not true. Maybe they're trying to pick up ultraviolet to synthesize vitamin B. There are a whole series of physiological possibilities that have been suggested in the mammalian and insect literatures. None of this do we have a clue. We simply do not know why they're coming out. We really need basic natural history data, observing what animals are doing, and accompanying with physiological studies to figure out if it's not feeding, why are they coming out? What physiological benefits do they get? So sort of putting this all together, it's clear that there are some major trade-offs associated with how deep into the soil an animal goes. Shallow depths are risky in terms of freezing, especially in very cold environments. The flip side is that they have the highest potential for activity. They'll get a cue that it's a warm day on the surface earlier in the day and can take advantage of that. And if the site is not too cold, they can have the lowest cumulative metabolic rate. Okay, how deep do reptiles go? Well, there are virtually no studies on this. No one has ever, very few people have ever tried to measure this. Amazingly, one person who did it was Cowles back in 1941. How did he do it? Okay, he noticed that in this area, they had these hummock dunes, but the farmers were flattening these dunes, scraping them, and converting this to agricultural land. So what Cowles did was follow behind these tractors, and when the soil or sand would kick up a snake or a lizard, he'd take its temperature, figure out how deep into the soil it was, and in the course of 4 days, he picked up 78 lizards and snakes. And here's a plot of where they were. Most of them were shallow. Okay. I think you can see why when I reread this paper a few years ago, I was absolutely stunned. It was so exciting, and Cowles first raised the issue of trade-offs, interesting physiological ecology, and then was able to find a way to study depth in sand. So the take-homes are that there are trade-offs involved with depth, but it's clear from a simulation analysis that there are lots of unknowns and many research opportunities available. There are lots of things that we need to study. These are all wide open, and I hope to be involved in some of these, but this is a major research area. So in ending, I would just like to thank quite a number of people. When I was a beginning graduate student, all of my heroes were older than I was, not surprisingly. As my career progressed, then I added heroes who were my contemporaries, and we were the ones who were coming up with new ideas and working together and exploring new things. But then as I got a little older, I started looking behind me, and there are all these younger people who were coming along doing just amazing stuff. And I admit to being competitive, but I'm just in awe of what the younger generation are doing, and very appreciative because it has helped keep me active for more than half a century in doing biology. So thanks to all of these people and many more have not listed. Thanks to Stephanie, Beatrice, and Diego and all of you for the invitation. And I hope you will find that winter is more exciting than I thought it was a few years ago. Thank you.
So thank you, Ray, for this amazing talk. I have some questions for you now. Let's start with this one, of Dylan Perez. To what extent does this variation in performance curves reflect adaptation to temperature? Can these curves be explained from a genetic basis?
Well, there are a number of studies now looking at the heritability of thermal performance curves or parts of thermal performance curves, and they are heritable to some extent. We've also done selection on parts of the curve like heat tolerance. And so yes, they do have a genetic basis. They can evolve, but typically they seem to evolve slowly. If you do comparative studies, generally you don't see major shifts within closely related species when you do a phylogenetic analysis. But they definitely can involve, especially the cold end, which seems to be more flexible and evolve more quickly. And there are empirical studies showing rapid evolution of cold tolerance in introduced populations, for example. So yes, there is a genetic basis. There's also a phenotypic basis based on acclimation, can shift these curves to some extent. Their maternal effects. So there are many factors that contribute to thermal performance curves, and also onto genetic shifts. So there is not just a single curve.
Okay, we have another one from Fernando Gomes. He asks, do you think dehydration would interact with temperature, determining performance curves and vulnerability to climate warming?
Yes, it's a very good question. There's a classic old study on toads where people manipulated temperature and hydration level. And you could show that how dehydrated the frog or toads were had a huge effect on how far they can jump. So that was the first example. It's now becoming clear, especially work that's being done in Australia out of Michael Carney's group, that water balance has a huge effect of interactions with temperature. Throughout most of my career, I think most of us tended to ignore moisture, but it's becoming increasingly clear that that has a huge effect. So it's probably temperature, moisture, the interaction between the two that will affect behavior and their ecology in important ways. So to the effect that climate warming is going to influence water balance, yes, it's going to have a big effect. And that's an active area of research.
Okay, another one, this is from Danilo Giacometti. Could thermoregulatory behaviors like microhabitat selection mitigate the negative impacts that climate change may have on growth and other traits?
Yes. So, for example, the example that I gave of the graphical analysis of the Sinervo et al. Proposal about reduced activity time, that's one of the behavioral ways that animals can use to modify their environment. You shift to an earlier time of day where it's cooler. You can also spend more time in the shade. You can find different microhabitats. Yes, that will reduce the impacts of climate warming, but it doesn't eliminate them, because in the process of selecting microhabitats or restricting times of activity, the animals have less time and less space available to them. So it can help, but I don't think it's a solution in many cases. Okay. And in some cases, animals don't have an opportunity to thermoregulate. Ones that, like in deeply shaded forests, the environment is homogeneous. There's really no opportunity for behavioral temperature regulation there.
Okay, thank you. Milene Gomes asks, could the digestion of food itself be one of the metabolic processes that would be impaired when temperature is too high for an animal?
Could you repeat that question, please?
Could the digestion of food be one of the metabolic processes that would be impaired when temperature is too high?
Possibly. I don't know. I do know that just the opposite is true, that low temperatures greatly inhibit digestion in reptiles. And that's, digestion is what is probably the most sensitive physiological trait I can think of offhand. In many animals like desert iguanas, which are normally active at say 30 to 40 degrees Celsius, if their body temperature drops below 28, they're unable to process food. They can can move down to 12 or 13 degrees, but digestion is very, very sensitive at low temperature. At high temperature, I just don't know. I suspect it is affected, but I've I don't recall seeing a study where someone has done that. It's an interesting question. My guess is that it will be impaired. High temperature is just bad.
Okay, that's true. From another one from Danilo Giacometti. Could the dual threshold idea for selected temperatures be also applied for the thermal cools lizards get while underground? So
I know the dual threshold, but I missed the second part of the question, sorry.
Okay. Could the dual threshold idea for selected temperatures be also applied for the thermal cools lizards when get underground?
Oh, I think I see what you're asking. The dual threshold model was that evolved. Originally, people thought lizards had a preferred temperature. It's very narrow. Then it was proposed, well, maybe they actually have an upper set point and a lower set point. So if temperatures are below this, they go bask. If they're above that, they head for shade. Within this range, they're quite happy and they just move around. So it's the idea that there are two thresholds involved. So yes, that's an interesting idea to apply to the soil. It could be that there's a dual threshold, as temperatures are rising, it hits one, and if it hits the other within a certain period of time, so there's, say something about the rate of change, they could then come out to the surface. The basic issue here, there are a whole series of potential models for a cue that a lizard would use to come out. You've just suggested one, which I hadn't thought of, which is a neat one. We used a simple threshold. It could be that maybe just a temperature change, it goes from cooling to warming. They could use a clock or some combination of all of the above. And there are just a whole series of possibilities, but nobody's explored. It's just wide open. And I think the fun thing about doing a simulation is because you have to parameterize it and make very specific statements or assumptions about cues that you discover, oh, this is something we don't know anything about. And I just think these are really exciting questions that can be explored.
Okay. Another one from Mila Pamplona. Is there any indication of seasonality in these animals? Do they keep their burrowing behavior to avoid freezing at constant temperatures in the months that are supposed to be colder?
We don't know. In terms of, do they change depths with altitude? Is that what the question is getting at?
I think so.
Yeah. There are very, very few examples where anybody knows what depth these animals are. Very few. Cowles' study, 1941, is by far the most comprehensive study of depths. And it's possible to figure this out. You put a data logger inside a lizard in the autumn, it will record temperatures every hour or however often you want. When it emerges in the spring, you decode that data logger. If you have temperatures in the soil at different depths, you can figure out very quickly how deep they were. Did they change depths? And you could do that for low altitude, high altitude, low latitude, high latitude, and start exploring these issues. It's really not been done. There are a few studies done on snakes at high latitudes in dens, where they're monitoring snakes move to a rocky den in the winter. A few lizards will do that as well. But basically, other than those den studies, there's nothing known, essentially nothing known.
Okay. Monique Simon, she asked, could the cue to come out be related with muscle physiology? Can they come out to maintain muscle health in some way?
Absolutely. There's Pete Zaney, who's done a lot of work on a lizard called Eudostansburyana. Pete thinks that the reason that they come out is for metabolic reorganization, so that they redeploy their energy resources, and that affects their entire physiology, but also their muscles. There's also possibilities of getting rid of toxic wastes that are accumulating over time, maybe activating immune function. There's just a whole series of possibilities, even vitamin D, as I mentioned. None of this is known. There's some studies for insects, but basically for lizards, there's nothing. And I think these are just really interesting questions. We do know in the Kalahari, as I mentioned, that lizards there, if they come out, they are feeding. So they are picking up some energy, but in other areas, they will come out and not feed. Pete Zaney's Utah in Oregon, it's a cold area. They come out, they bask, they do not feed. Why? Why are they coming out? We don't know. I just find that these are such fundamental natural history questions that, and physiological ecology questions that we have not explored. And I think the reason is that most people thought the same way I do. Winter is boring. There's not much happening. It's more productive to spend your time back in the lab, analyzing data, writing papers. Why go out and look at the snow? I mean, it's just, it's boring. I was wrong. I find this a really exciting set of opportunities.
Okay, Milene Gomes, is there any field observation about how often lizards change the depth at which they underground? Is it possible that they change that depth during the day, for example?
It is. There's very little information on this. There are one or two studies which show depth changes over time. If an animal is simply buried in the sand, it's going to be very difficult to change surface. If it's in a burrow, then potentially it can move up and down easily. We do know that snakes and lizards in rocky areas where they go into the crevices in a rock, they can move up and down during the course of the day or change their depth seasonally. But for lizards like in Cowles areas, we simply do not know what they're doing. In the paper, I did do a series of analysis allowing an animal, most of the analyses were done for animals at fixed depths, 5, 10, 20, so on. But I set up models where the animals could move and they could always go to the coldest place in depth. So they could change depth easily at no cost and simulated how much better they could do. And the answer is not much, most times. But these are fascinating questions. Again, they're just basic natural history. And you can find a few papers, one or two places where somebody has put a transmitter and been able to determine whether or not an animal is changing depths. But by and large, we don't have any idea what they're doing.
Okay, we have a long one, so please listen to me carefully. From Rafael Bovo. For a long time in ecological physiology, many researchers have been using the assumption that locomotor performance has a close correspondence with fitness. However, different activities have different optima. Also, the correspondence performance fitness might differ among ectotherms. Could you say more about this issue in a historical perspective in the literature? Do you think this framework could change in the next decades?
Okay, this issue came up fairly early. I remember in 1982 when I published a chapter in Biology, the Reptilia on this. One of the issues I raised was different optimal temperatures for different traits. And there's a figure in there that shows that. In most cases, in the lizard data that we had at the time, the optima were fairly close. The curve shapes were different. For example, if this is a show for locomotion, for digestion, it was more like this. So as I said earlier, digestion is very temperature sensitive and shuts off at low. And why? Why? The ideal world would seem to have everything have the same thermal performance curve, the same optimal temperature. That would seem a priori to be ideal. But there are differences. Okay, then now what do you do? Well, I think there are two kinds of questions. One is, are these merely necessary because of the way digestion works, that it's more temperature sensitive than, say, locomotion? And there might be some mechanistic reason that I don't understand. The other is to say, maybe these are evolved differences. Maybe it's advantageous to have digestion occurring only at a high temperature and not at low. And to my knowledge, no one has ever really tried to model this and try to think it through. It's a question that has come up. I mean, it was, as I said, it was a question in the late 70s and early 80s, periodically pops up. And I think it's unresolved, at least unresolved as far as I know. We do know that some traits have different thermal sensitivities than, say, locomotion. But we don't know whether this is purely mechanistic. You can't make hearing have the same thermal sensitivity as digestion or locomotion, or whether these are adaptive differences. I think it's a really interesting question. Periodically, I've thought about trying to get involved, decided it was hard, and have never really grappled with it. And I should, because I mean, I've been aware of it for over 40 years that this is an issue. I haven't had an insight that would allow me to jump in and figure out that I could solve this or make progress towards it. But it's a really interesting question.
Okay.
I wish somebody, maybe you will do it.
Maybe.
Yeah.
Carla Mother asks, what advantage do you see on investigating thermal performance in different levels of organization, such as the cellular level?
When I was a grad student and first starting to say, which level of organization did I want to study? Whole organism, tissue level, cellular level, molecular level. I made the decision to pick a whole animal trait because I was asking ecological questions. So I wanted an organismal level trait because that's closest to the animal in nature. And I felt that a lower level thing like tissue performance or activity of enzymes, something along those lines, was just way too far removed from the whole organism. But as George Bartholomew emphasized many years ago, and incidentally, for those of you who've never read papers by George Bartholomew, he was one of the great comparative physiologists. And in the 1960s, in a series of papers, he laid out the philosophy of evolutionary physiology beautifully. And one of the points that he made was that there are multiple levels of organization, and each level derives its significance from the next level above and its mechanism from the layer below. So I can measure the thermal dependence of locomotion. The relevance of that is to the ecology of the animal. But you can also go down and look at how the muscle firing works at different temperatures or the enzymes that are involved. And that gives you a mechanistic explanation for why the lizards run the way they do. You really want all of these. But since I was an ecologist and I was asking ecological questions, I went to the whole organism, not to the lower levels. But those are important mechanistic issues, but I think they're too far away from the ecology to, I don't think you can take the thermal dependence of an enzyme and predict the running speed of a lizard in nature. Don't think you can do it. Why not just measure thermal dependence of running speed directly at the whole animal? But you do want to understand the mechanism. That's also important.
Okay. Rafael Lara, he asks, about hours of thermal restriction, what do you consider is a better threshold? Preference temperature range or voluntary thermal minimum maximum?
I think those are the same. Well, could you repeat that again? I think I misunderstood the question.
Okay. About hours of thermal restriction, what do you consider is a better threshold?
Oh,
Pref range or voluntary thermal minimum maximum?
okay. Hours of restriction was a term that was coined by Barry Cinervo and colleagues in that 2010 paper. And the idea, the ecological issue is with warming, the animal has to restrict its activity time. And if the hours of restriction, or conversely, the hours of activity, if the hours of restriction grow very large, then this can lead to reduction in food and energy balance and lead to extinction. That was the basic idea in Cinervo et al. The question is, what do you use to measure hours of restriction? And I think that's a good question because you'll get slightly different answers. If you use the thermal preference range, it's probably narrower than the voluntary maximum and voluntary minimum, and you will get different answers. I think I would rely on what do we know about the behavior of animals in nature, and I would look to see what temperatures do they accept for activity and use those. You could do it empirically that way. I think if I had to work without direct information from the field, I'd probably use the thermal preference range because it's narrower and that's what the animals are using. If you had, going back to an earlier question, if you had a dual threshold model where you knew the two set points, that would probably be a pretty good one to use. But that's known for very few species. Or, when in doubt, when you have to make a decision how you're going to parameterize a model, do it both ways. Use thermal preference, use voluntary maximum minimum, see if you get the same answers. If you do, then you have a pretty robust result. It's basically a sensitivity analysis. So I guess what I'm saying is it may not be either or, try several different possibilities, see if you get the same answers, hope you do. If you don't, then you have to get more information from the animals themselves, what they think is important. And that requires a field study.
Okay, there is another one from Rafael again. According to your model, how will global warming affect fossorial species? For example, Bipes or Aniella?
That's an embarrassing question. I haven't thought about that. And the reason it's embarrassing, that one of the projects that I did in the Kalahari many years ago was on fossorial species. So I spent a lot of time digging for lizards underground, and obviously should have thought this one through. But thank you for suggesting this. I will start thinking about it. For a quick answer, with climate warming, the surface temperatures will get hotter. Which will mean if the soil temperatures are sufficiently hot, the animals have to go deeper into the soil. And we've started looking for soil temperature data changes over time to see what the nature of the patterns are. Is it warmer at all depths or just primarily at the surface depths? We really haven't explored that yet, but it's an interesting question. There's a whole world of organisms that live underground. And of course, for plants, their roots are underground, and it's going to affect, the soil temperature is going to affect root and root physiology as well. So yeah, it's a really interesting question. And I don't know, right offhand, I can't think of anyone who's explored this or commented on this in the literature. There may be, someone may have done it, but I just, I'm not thinking of an example. But it's, soil temperatures will get warmer. And if they are too warm, that will force animals down. For some organisms, warming soil temperature will actually improve conditions. So in cool areas, they may be able to invade areas that previously were too cold for them to survive. So I don't think there'd be a simple answer that's applicable everywhere. But if it's hotter, they have to go deeper. Hopefully the things that they are eating are also going deeper with them. The fossorial lizards in the Kalahari are termite specialists. Something like 98% of their diet is termites. So what the termites do in response to warming will have a big effect on those lizards.
Okay, thank you. A question from Aníbal de la Vega. Do you think that microbiota could play a fundamental role during the cold season to wake up?
So the microbiota,
Yes.
it could well. People are just starting to explore the microbiota of reptiles. So I think it's premature, but it's certainly going to affect what's there, the relative numbers, different species. But this is not something that I've worked on. I will say that if I were starting my career right now, I think this is a general area that I would go into, the interactions between microbiota and organisms, because it has a huge effect on their physiology. I mean, when I started, my worldview that there was a lizard, it had a physiology, I could understand it. Now it's increasingly clear that a lizard is a community of organisms. And what this lizard does may depend not only on its own genotype and its own background, acclimation, but also very much on the organisms that are inside it or on it. And that makes, that's a real complication that has only become apparent in the last, I think to most of us in the last few years. But I find that really exciting. It's upsetting because I like to think that's a lizard, I can understand its physiology. It's to view it as a community of organisms is, I think, really exciting. New direction that people are exploring.
That's true. I have a last question for you. Do you think that the threshold of body temperature that triggers sheltering could change across the seasons or the environmental temperature?
So so this would be, say, the maximum voluntary temperature that if an animal reaches that and it heads for shade or shelter, does that change seasonally?
Yes.
Okay. It might well. I'm not. Empirically, we can go to lizard data that we have in the Kalahari, and we would see undoubtedly that the highest temperature accepted in summer is higher than in winter. But is that a change in the behavior of the lizard, or simply that during the winter it never gets hot enough for the equilibrium temperatures to reach that? So I don't know of any study that's quantified that. I wouldn't be surprised there's seasonal variation. There's certainly a seasonal variation in some species and things like thermal preference. There can be seasonal variation at critical thermal maximum. Given that, I would expect that there is seasonal variation in the upper threshold temperature for seeking shade. I just can't think of an example right offhand, but almost certainly it's going to occur in seasonal environments.
Okay, then. So with this last question, I think we can end today's section. Thank you very much once again, Raymond, for the wonderful talk and this wonderful discussion. We really appreciated that. I would also like to thank everyone who watched us and stayed here until the end. And I hope you enjoyed this transmission as much as we did. And we'll be waiting for you all once again for next talks. Thank you very much for everyone. Bye-bye.
Bye. Thank you.